Hi students, today we are going to discuss molecular basis of inheritance. This is a chapter with high weightage as we all know, and you have to study this chapter very well for your board examination. More number of questions as well as more marked questions you can expect from this chapter, and it is not easy to complete this chapter within 10 minutes or 15 minutes or half an hour. So I will give you a bird's eye view regarding this chapter, the concepts from where the previous year's questions being asked. Along with that, some of the basic concepts which should be there in your mind before studying this chapter. So, shall we start? Okay, so this is, we are learning regarding DNA. So, where is the location of DNA? It is inside a cell. So, we know that cell, inside the cell nucleus is there. Inside the nucleus, chromatin network is there. Inside the chromatin network or the chromatin network during the time of cell division converted in the form of chromosomes. And these chromosomes are made up of DNA. So a piece of DNA which codes for a single character is known as gene. This is the basic concept. And then we know that we have mentioned DNA. It is the genetic material of majority of the organisms. So DNA, deoxyribonucleic acid. So it's a nucleic acid. So what are these nucleic acids made up of? Which are the two types of nucleic acid, DNA and RNA? And we all know that it is from the RNA, DNA is formed as a result of evolution. It is there in the textbook, the functions of RNA. There are specific functions are given in the NCERT textbook. Go through that. So nucleic acid. Nucleic acids are made up of nucleotides. They are made up of nucleotides. And in turn, nucleotides are made up of nucleosides and phosphoric acid. Nucleosides are formed of nitrogenous bases and sugars. And which are the nitrogenous bases? Purines and pyrimidines. Purines consist of adenine and guanine. Pyrimidine consists of thymine, cytosine and uracil. Sugars, ribose and deoxyribose. Ribose sugar in RNA, deoxyribose sugar in DNA. This is another basic concept. So now we understood what is a nucleotide. So this is the structure of a nucleotide. We have already learned a sugar is the centrally located 5-sided sugar is there, and then phosphate group is there, nitrogenous base is there. So such kind of several nucleotides are formed to join to form polynucleotide. Its picture is there nearby that nucleotide, a single nucleotide. So polynucleotide, DNA is a polynucleotide. And which are the different bonds present in this polynucleotide? Usually that question is asked for maybe for 1 mark or 2 mark. What are the different bonds present in the DNA or this polynucleotide chain of DNA? There are 3 bonds. The bond between the nitrogen bases, it is hydrogen bond. That is number 1. And then the bond between the nitrogen base and the sugar, N-glycosidic linkage, then phosphodiester bond. These are the 3 bonds which is present in the polynucleotide. Then comes central dogma, which is proposed by Francis Crick. In this chapter, you are getting a lot of scientists' names, so that you have to study. Large number of a number of scientists' name is there in this chapter that you have to study without leaving. Can be asked for your MCQs. So here central dogma, this is all about this chapter. Central dogma, even though it is in one line, it describes the entire chapter of molecular basis. So central dogma, which was proposed by Francis Crick, according to this, DNA can undergo replication. That means it can make a pre-existing DNA can make a new DNA. That is DNA replication. And then the next is about protein synthesis. The first step of protein synthesis is transcription. Transcription means A DNA contains all the code for making a protein. And we know that proteins are nothing but amino acids joined together by peptide bond. So there is a specific order for this amino acid to join. And that code is there, that order is there in the DNA. But DNA cannot directly synthesize protein, or they cannot combine this amino acid to make the protein. So what DNA is doing? The DNA is giving that DNA is transferring that codes to the mRNA, or from the DNA an mRNA is making with all the codes. And this mRNA can produce or can actively participate in the protein production. So from the DNA, mRNA is produced, and that process is transcription. From that mRNA, a protein is producing, and that is translation. So this is actually the entire chapter is all about. Now we will move to the how the DNA is packed in a cell or in a nucleus. It is in the form of nucleosome. This diagram can be asked for the examination. Even though in the first diagram you can see a single ball, it is not a single ball. It is actually histone octamer. 8 units of histone protein is there. So basic nature of histone protein is due to 2 amino acids. Which are they? The answer is there in the NCERT textbook. Find it out. And you can see enlarged version of the histone and different histone octamer. Around that octamer, DNA is wounded. You can see that. Then this is the histone and DNA. And now we are moving to DNA as the genetic material. There are 3 experiments. There are a lot of experiments in this chapter, but the 3 experiments are explaining how DNA is found as a genetic material. So search for genetic material. The first experiment we have to study is transforming principle by Frederick Griffith. You can see this diagram itself explains how the experiment is all about. So which all the questions can be asked from this area, like illustrate or explain the experiments conducted by Frederick Griffith in order to prove that DNA as a genetic material, or else explain transformation principle. That is also the same thing. Or sometimes 2 more questions such as which are the organisms Frederick Griffith has taken to prove DNA as a genetic material. So you can see here how the mouse is living, how the mouse is dying when different strains of bacterium, Streptococcus pneumoniae bacterium is applying or injecting into this mouse. Do study this experiment very well. Okay, so rough strain is there, smooth strain is there, heat killed smooth strain is there. And what all changes are happening whenever these different strains are injected into the mice, you can see here. And at last, rough strain and heat killed smooth strain together injected. At that time also what happens, that explains transforming principle. This is nothing but the bacteria. There is a certain, at that time Frederick Griffith was not identified the DNA. So some material from the bacterium is transferred to the another bacterium. That property of that bacterium is known as transformation. That is what is happening here. And then now the next experiment comes by Avery, McLeod and McCarthy, chemical characterization of transforming principle. They have taken 3 enzymes, DNase, protease and RNase enzyme in order to explain or in order to find out the reason for the transforming principle. And they found that with the DNA is the genetic material. How they found out? Here also the whole answer is there in this diagrams. So the first diagram, you can see that, they are adding proteinase to the heat killed cells, then RNase, then DNase. And whenever DNase is added, that time no transformation happening. That means DNA is the DNA is the genetic material. And then comes the next experiment which was done by Hershey and Chase. This is very important experiment. So many times it has been asked for the examination for 5-mark question as 5-mark question. So here also the diagram is there to explain the entire process. You can see that the bacteriophage, they have used bacteriophage, and S strain and P strain. Radioactive sulfur, radioactive phosphorus they have used. This can be asked for 1-mark question. Which radioactive element is used in this particular experiment done by Hershey and Chase? P32, S35. And all the 3 points are there. The 3 steps are there: infection, blending, and centrifugation. I'm not much explaining the experiment. You do study this experiment. This is very important. So 5-mark question you can expect from this. And then DNA replication. DNA replication, already definition I have given. And here also Michelson and Stall's experiment, very important. From this experiment, either directly experiment can be asked, or we can expect such kinds of experiments in the section D. Because sometimes the diagram will be given. On the basis of the diagram, 3 questions, as we all know in the case-based question. So here also Michelson and Stall's experiment, which explains semi-conservative method of replication of DNA. And a semi-conservative method of replication means one of the parental strand will be retained in the daughter colonies. So here they haven't used any radioactive, instead heavy isotope of nitrogen, N14 and N15 they have used. Okay, so hybrid DNA they have produced. So how much hybrid DNA is produced in different generations? Such kinds of question. How much is produced in the 4th generation, at the end of 4th generation? Or what is the ratio of the hybrid versus normal DNA? So such kinds of question you can expect from here. This is very important experiment. Next is about the replication. From this 3-mark question you can expect a replication fork is given here. A Y-shaped fork. Which all are the enzymes involved in the replication of DNA? For unwinding the DNA helicase. For starting the replication, RNA polymerase is needed. And then a DNA polymerase for, the production of DNA. And then a cutting enzyme, restriction enzymes are needed. Then the joining enzyme, DNA ligase are needed. So likewise, you have to study the replication. Continuous strand is the non-continuous, discontinuous strand is the leading strand, is the lagging strand is the. Why it is leading? Because the peculiarity of the DNA polymerase. What is that peculiarity? Find it out. And which is in the leading strand, which is the template strand. What is its, whether it is 3', 5' or 5', 3'. It is 3', 5'. Okay. So such kinds of question you can expect from here. And then comes transcription. As I have already mentioned, transcription is the production of mRNA from one of the strands of DNA. Why in DNA both the strands are not involving in transcription? Answer is there in the NCERT textbook. Find it out. Okay. Initiation, elongation, termination. 3 points are there clearly in the transcription of DNA, and transcription in prokaryotes as well as transcription in eukaryotes we have to study. In transcription of prokaryotes, I have mentioned initiation, elongation, termination, 3 distinct points with the diagram we have to learn. And transcription in eukaryotes, the major point is RNA splicing, or we can say that post-transcriptional processes. After transcription, in eukaryotes, certain processes should be there, and that is RNA splicing, introns and exons. Introns are intervening sequences, that means non-coding sequences that we don't need in the mRNA, processed mRNA. So introns has to be cut and removed. Exons are the expressed sequences that we need for the mRNA. So in a eukaryote, in the mRNA produced after transcription, it consists of introns as well as exons together. So introns has to get removed, exons need to get joined, and this is actually known as RNA splicing. Okay, and after splicing, the spliced mRNA should be identified. In order to identify that, there is a process of capping and tailing. Capping is with 5-methyl guanosine cap and polyadenine ring. Okay, so that is it. And then comes the genetic code. Genetic code is very important. Which are the initiator codon? Which are the initiator amino acid? What is triplet code? So many scientist name is associated with that we have to study. And then then that is about genetic code. Some of the important characteristics, features of genetic code. Code is degeneracy. Degeneracy is one of the important character. What is that? What does it mean? 2-mark question. It means for coding one amino acid, more than one codon is there. More than one codon can code for one amino acid. So that is it. So likewise, 7 or 8 salient features of genetic code is that we study. And then comes the tRNA structure. Structure of tRNA very important that we have to study. And anticodon site, UAG. Here you can see the anticodon. AUG, we'll read the AUG, which is the first codon of the mRNA that will code for methionine. AUG has got dual function. What are they? It is the initiator codon. It codes for the amino acid methionine. These are the 2 dual functions of AUG. And then how translation occur, the process. It consists of so many stepwise process. All the steps we have to study. Very important for 5-mark. Translation. So translation, the first thing is tRNA charging, amino acylation of tRNA. Then ribosomes are coming and joining with the mRNA to that initiator complex. Now the process is starting. Okay, so all those things we have to study. Next comes lac operon. Before coming to lac operon, translation, you have to understand one more thing, termination. Initiation, elongation, termination. Termination, which are the terminator codons? UG, AU, AG, UAA. These are the 3 terminator codons. And what are the peculiarity of the terminators? It is given in the NCERT. Try to study. And then lac operon, operon concept. Who introduced the operon concept and what is operon? Operon means it's a set of genes which is controlling a metabolic reaction. Okay, so lac operon means lactose operon, which is happening in the bacteria. So what is it? It is actually lactose metabolism. It is a set of genes which controls the lactose metabolism. Here 2 metabolisms are there. Here 2 points are there. Switching on of lactose metabolism, switching off. That means whenever the lactose is there in the system, the enzymes which is needed for lactose digestion is produced. Otherwise, no need of producing that enzyme. So whenever lactose is there in the system, enzyme should be produced. Whenever lactose is not there in the system, we don't need the enzyme. So that is switching on and switching off. So lac operon is very important. You can expect this for your examination as 5-mark question. Even the diagram. With the diagram, you can easily explain it. So lac operon means, as I mentioned, suppose it is switching on. That means lactose is present there. So if lactose is present, what will happen? It will go and bind with the operator region of the operon so that RNA polymerase can easily bind with the promoter region and the 3 enzymes are produced. Which are the 3 enzymes? 3 enzymes are beta-galactosidase, trans-acetylase, permease. These are the 3 enzymes. These enzymes are produced by the genes which we are calling as structural gene. Each of these enzymes are produced by specific structural gene. You know that it is ZYA. Okay. ZY, structural gene Z, structural gene Y, structural gene A. Each of these genes producing the enzymes such as trans-acetylases, beta-galactosidase, permease respectively. Beta-galactosidase is the enzyme which breaks down or digests our lactose. Okay. So that is it. If suppose lactose is not there in the medium, we don't need these enzymes. So what happens? There is a specific gene called regulator gene, and that will produce repressor mRNA. This repressor mRNA produces a specific protein called repressor protein that will come and bind with the operator region. So that operator region is blocked. So RNA polymerase cannot come and start the process of this production of enzymes. Okay, so this is happening in the switching off. So this is actually the lac operon, and then comes DNA fingerprinting. DNA fingerprinting, which is also very important. The diagram, the entire process is given in the diagram, and after that the explanation, the points also I have given here. You can see what are we doing in the isolation, in the DNA fingerprinting. So which is the first step? Isolation of the DNA. Then polymerase chain reaction, PCR, in order to increase the number of DNA, number of sample, we need more DNA. Then cutting with the help of restriction endonuclease, gelilactophoresis for separating the fragments of DNA by using certain dyes, and that dye's name is important. The under UV rays it is observing and it appears as orange colored bands. All these are very important. And this double-stranded DNA we are making into single-stranded DNA by using certain alkali. Which all are the alkalis that you have to find out. And then the process of blotting. And then a nitrocell blotting means it is converted or it is transferring into a nitrocellulose membrane. And then DNA probe is introduced. One more question. What is DNA probe? It is a single-stranded DNA of non-nitrogen bases. We know the the nitrogen bases in that DNA, so that it single-stranded DNA plus this DNA probe will bind together and a hybrid is formed. Autoradiography, and we'll get the DNA fingerprint. Okay, so almost all the points I have discussed here. You can see, 2-mark and 3-mark questions are asking from these areas. So many problems are also there in this chapter. Please go through the problems. 5-mark questions are here. You can see, go through the 3-mark questions. The second question is directly from the NCERT textbook. Even the same question is there in the NCERT textbook. Dual function of codon I have explained here. And this kind of question you can expect. Given below is a part of template strand of a structural gene. And transcribed mRNA, how you will write this transcribed mRNA. Replicated, replication also, such kinds of question can be asked for replication type also. Replicated DNA you have to find out. So here I haven't given the 3', 5', that also you have to mention in your answer script. 3', the polarity of the DNA also you have to mention. 3' to 5' or 5' to 3'. Okay, so this is different questions. You can go through the questions. These are the pattern, unambiguous, universal, degenerate, some of the terms. Explain each term. So it is directly there in the NCERT textbook. Find out. And need label the sketch of replicating fork. You can see here. See, all these are the previous year's questions. See, 5-mark questions. You can see here. All these are 5-mark questions. See, this kind of question can be, I already mentioned for section D, this can be asked. And see, all these are the different questions from the previous year's question paper. So, study very well. All the very best. Thank you. Bye.